part of ours. We need to get the components of the CRISPR-Cas9 into these human cells. How would you do that? One way that this gets done is by creating a
Is it the
plasmid
same person? Yeah. Is it Carzon?
that contains the DNA sequences that we need. What would those be?
Bro, I need to answer.
What would those sequences be? What should the plasmid have if we want to use the CRISPR-Cas9 system to base edit human DNA? We're going to take a system from bacteria and use it in humans. One way of transferring the genes is to use a plasmid. What are the genes that will be part of this plasmid?
I know. It's okay.
What do we need? Go on, one of them's really easy. It's going to need an origin of replication. Yes. What else is it going to need? Think about what the CRISPR-Cas9 thing is, right? What else do we need here? Sorry, what do we need? So what is that CRISPR RNA called? No, not that. So for our purposes, right? For our purposes, I forget which piece of paper I wrote this on. For our purposes, this is our guide RNA, and it's been simplified. We are just going to have a single RNA molecule that's going to make up part of the effector ribonuclear protein. But our cells are not making this, right? So somewhere in here, there has to be the sequence that is going to produce our guide RNA. Remember, this guide RNA is going to take the endonuclease to the right place to cut the sequence. What else do we need? Oh, we need that to be expressed, so there's going to have to be a promoter there, right?
Do we need restriction enzymes for something to be cut?
Everyone wants to use restriction enzymes,
No.
but I'm talking about the plasmid itself. Let's just act like it's all been put. The way that it will have been assembled is with the use of restriction enzymes and ligase for sure. But let's think about what would it look like once it's all been put together. What else do we need? So no, I don't think that we're going to need an operator locus here.
Ampicillin?
So we are going to need what we may not have to use ampicillin, but in this particular, so let's put ampicillin resistant here. The reason that there's going to be an ampicillin resistant gene is because this thing will have been assembled in bacteria. Only once it's fully assembled will it be transferred over to eukaryotes. And so ampicillin's a good one. There will be another one which enables you to tell whether eukaryotic cells have been transfected, they've gotten the plasmid or not. Where neomycin is really common, but yeah, we'll have that. What else?
Do we need the GAG sequence in the plasmid?
The what?
The GAG sequence?
Yes, we have to have the template sequence. One possibility, so that sometimes that gets introduced of it, but we need our template that's going to be used during the homology-directed repair process. That can be part of our plasmid as well. What else? There's an obvious gene missing. The which? So these are going to be our two selective markers, one that we can use in bacteria and one that we can use when they're in eukaryotic cells. So this template. No. You're really on the right track here. This is the gene of interest.
That's what I'm talking about.
And this would contain the correct sequence. Yeah. Here's the RNA component. There's another obvious component that's missing.
You see that?
Yeah, how about the endonuclease, the Cas9 protein, the Cas9?
So let me see.
These are the basic elements, right? This is a complicated process. It's a complicated process to do this. Fortunately, you don't even have to do it yourself anymore, right? So you can buy this. You can buy this. So you're gonna here's the Cas9 gene. This particular one has, as an extra reporter, has this thing called green fluorescent protein. It's a super easy way to find out whether cells have taken up the plasmid because they will literally glow green under a light. But you can see there's the guide RNA, there's promoters for the things you want. That the PUC is an origin of replication that came out of a plasmid. Kanamycin, it's just the stuff you want. But for $800, you can do this, and you'll have your plasmid sent to you. That black box there is where you're going to supply your own gene, though. I think that's what the black box is. But they'll send you the whole thing pre-made.
This one. How do I select the first?
How are we going to get this into eukaryotic cells? How do you get this? Sorry? One of the best options is to use a viral vector, a virus, to carry this into the eukaryotic cells. But this is pretty hit or miss, right? You obviously when you took these stem cells out of the patient, you have a lot of stem cells, but not very many of them are going to be edited. Not many of those will be correctly edited, right? You're going to have to go through bunches of steps after that. You're going to have to take individual cells, grow them up as clones. You're going to have to sequence the gene. Did I make the edit that I wanted to make? And only after that would you have a group of cells that you could say, yes, this is a group of cells that I can transfuse back into the patient to try to give her the ability to make her own hemoglobin. What is amazing is that they've actually managed to do that now. There's a woman who had just terrible sickle cell disease, and she's completely cured of it because this whole process went right, and now she just makes her own normal hemoglobin. the whole procedure costs like, I think they said it costs like $2 million. So
I took 1, 2, 3,
so
4, 5.
they've done a cost, I heard about a cost analysis of this for actually another related blood disease in England. And the National Health Service in England has decided it's cheaper to pay $2 million per patient than it is to continually have them in hospital needing blood transfusions and other kinds of intense treatment. So lifetime cost, it's actually cheaper to be able to do this. The reason it's $2 million, right, is that this is really it's done for just a single person. You take her cells, you edit them with CRISPR, you hope, you transfuse them back into her, but her cells wouldn't help another patient because another patient would reject that. It would be a tissue transplant, right? It would go through rejection. So this whole system has to be done one patient at a time, which is going to always make it really super expensive, but apparently worthwhile when you cost it out versus the rest of the care. Okay. Let me say a couple of other things about CRISPR. No, actually, yeah, let me say one other thing about CRISPR and one more thing about cloning. Number 3. You will remember how much of a fuss I made about the benefit Mendel had of being able to go down to the seed store and pick peas that came in 2 different strains, round or wrinkled, yellow or green, right? When you're doing genetics, that kind of variation is typically not available to you. So we're in a very different situation now, right? We have lots of DNA sequence data, we know lots of genes, but we often don't have that kind of variation which really helped Mendel understand inheritance. The question now, of course, is not the pattern of inheritance. The question is more like, okay, so that's a gene. Well, what does it do? What is the function of that gene? One of the easiest ways that we can address that question now is to use CRISPR to knock out the function of that gene and then look at the phenotypes. We're effectively generating now, we're generating the kind of variation that Mendel could just go down to the store and buy. So we can actually generate mutants. So this is an easy thing to do in comparison to the base editing. All we need to do is cut the DNA and allow the non-homologous end joining, which creates insertions and deletions. This is really likely to lead to loss of gene function. Ideally, you'll breed the animals. Let's say we did this in mice. We're not going to be doing this in humans, obviously. Let's say we did this in mice. We breed the animals so that we see homozygotes, and we can look at the phenotype. This is a super, super common way of trying to figure out what genes are doing. You basically knock out their function. Now, because CRISPR is, it's not 100% efficient, you're not going to knock out both alleles in an individual cell. But you then breed the animals until you have homozygotes, and then you see what was that gene doing for those particular animals. So this is a really, really excellent use now for CRISPR. My last thing. one other use of cloning. So, what do you guys know about insulin?
Diabetes.
Yeah. Diabetes is required to help break down
That's
the
where I'm headed.
glucose. Break down the
What'd
glucose.
you say?
Or build. Is it break down the
It's a
glucose?
secret.
Okay.
I don't know. I just said diabetes.
It's required to like break it down.
So what kind of molecule is in insulin?
Beta.
Sugar.
No,
Is
it's a beta.
insulin a sugar?
No.
Does it end in OSE? No.
It has a beta something.
It is a hormone.
Wait, I know this.
It's a beta something.
Wait.
What kind of molecule is insulin?
Oh my god, it's so.
I should know this, bro.
Everyone's heard of insulin, right?
Oh my god.
It's a protein, it's a peptide.
You should have said it out loud. Yes, you should have.
51 amino acids. There are lots of these, right? Growth hormone. Growth hormone's longer. It's also a peptide, but it's somewhere around 200 amino acids long. These things are really useful, right? Do you know where insulin used to come from?
Pigs.
It came from animals. Yeah, people extracted it from pigs, right?
Yo,
It's produced-
just take the test now.
right? It's produced by the adrenal glands. They used to take those out. They used to purify the insulin, which is very similar to our own insulin, minor differences. And that was what patients had, right? Access to. Nowadays, we can actually get human insulin, and we're not taking it from other people. We're producing it pharmaceutically. Similarly with growth hormone, right? Growth hormone, some people are born, they're not producing enough growth hormone, so they're not, like little kids, they're supposed to be a certain size at a certain age. So kids can be given growth hormone to accelerate their growth. And athletes can use growth hormone because it makes people bigger, but it's unclear whether it makes them stronger. Anyway, it's also a drug of abuse.
What's that?
But growth hormone is the one that I wanted to talk about, right? So, growth hormone is this fairly long peptide. The gene has 5 exons.
Oh,
They also show alternative splicing. So the 200 is the full length normal form of growth hormone, but there are other forms of it in our body. If you wanted to produce growth hormone pharmaceutically, what would you produce it in, right? You literally cannot go around taking out people's pituitaries. They need them, right? So we need to be able to produce it somewhere else.
Monkeys.
Where should we produce it? How about we produce it in bacteria? What would it take to get bacteria to produce one of our proteins?
You get some viruses that transport human DNA into the bacteria.
So that sounds a little chancy, right? We want it to be this particular gene.
I would too.
Oh,
Without stress
we
management,
could
it's
make
13.
a plasmid. I told you this would be getting back to cloning. We would make a plasmid. That plasmid's going to have an origin of replication. What else is it going to have?
Promoter.
The GRRNA.
The promoter.
Huh? Are we going to use the laxi? No, in this case, we don't want to use the laxi protein.
Promoter region.
What do we need to be put in our plasmid? We need the gene for human growth hormone, right? But
I took 15 spoon. But I just,
where are those? Here we go.
I think close to 18 scares me.
What is the problem of just taking the gene out of a human, cutting it out with restriction enzymes, ligating it to a plasmid, and then getting that gene into bacteria? It would still have introns, and bacteria not only don't have introns, there's something else about them that they don't have. They don't have the splicing apparatus. But bacteria, that spliceosome is in our nuclei, but it's not in bacteria, right? Why would they need one, right? This gene has these 4 introns, 5 exons, right? And if you took the gene and put it in bacteria, it'd be completely useless because the coding regions would be interrupted by all of this junk, which couldn't be spliced out. What should we do?
Splice it first.
We could engineer what?
A nuclease.
Well, so nuclease would be too tricky, right? Remember, the spliceosome is this incredibly complicated machine, right? There's 5 of those small nuclear RNAs, and then there's over 100 individual peptides
It's my
also.
skin color, but it's still boring.
So engineering that just too much.
It's
Integrate it into the plasmid after the mRNAs.
Oh, yes! This is it. This is it, right? You don't take the gene itself.
My mom does.
We take the mature
She doesn't.
messenger RNA. The mature messenger RNA has already had the introns removed. But we can't ligate RNA to DNA, right? This is a single strand molecule, and we can't ligate it into this. So what should we do as an intermediate step?
Cheese protein.
This is something that, for example, the single-strand virus, HIV, also does during its life cycle. It's a single-strand RNA virus, but it gets integrated into our genomes as a piece of double-strand DNA. How does that happen? Yes, there is an enzyme which HIV carries with it when it infects cells called reverse transcriptase. It's called reverse, transcription, right? The transcription is just making an RNA copy of a DNA template. A reverse transcriptase makes a DNA copy of an RNA template. So then we have a DNA copy. But this DNA copy will have no introns. And that is going to be the thing that we want to clone. What else are we going to have in our plasmid here? Anything else that's parallel to the previous? We've got to have a promoter, right? it is essential that this gene get expressed, right? When you're cloning into a normal plasmid and you're just trying to characterize DNA, where I started this discussion of cloning, it really doesn't matter whether there's a promoter there. You're probably going to cut it up with restriction enzymes. You'll do some DNA sequencing. You're not interested in this process of expression. But here, we want to express this particular plasmid. And then there's one other thing that we want to, one other element we want to include that came up in previous plasmids.
Ampicillin.
We want some kind of marker to know whether the cell has the plasmid, right? So in eukaryotic things, it's often the gene for neomycin resistance, which is useful for that thing. But it's quite possible, you saw on the diagram that I showed, that you might include the green fluorescent protein to know that the cells do indeed have this particular thing in them.
Aren't we putting
Oh,
this?
yeah, we're not putting it into
Yeah.
eukaryotes, we're putting it into prokaryotes, aren't we? So we can use ampicillin resistant. Good point.
I
Good
didn't
point.
even say anything. You said
Yeah, no, that's a good point. And then we can express this, and this is how we produce insulin now. We don't get it from pigs. It can be produced pharmaceutically, which and it's been in the news, right? You can then rip people off for it. It's really really cheap to produce, and you can charge $200 for it or something. But human growth hormone, the same thing. We just produce these things in bacteria now. It's this relatively simple process called cloning. Obviously, there are technological details, but it's a relatively straightforward process.
With reverse transcription, you were talking about how it's an HIV-infected bacteria. Could we do reverse transcription normally, or do we need to introduce something?
No, we need to use the reverse transcriptase enzyme. Yeah. So, and my guess is that I bet the one that we use commonly came from HIV in the beginning. We do not normally in our cells make reverse transcriptase, but we have the genes for it, which are these ancient retroviral elements. They are one class of those interspersed nuclear elements, are ancient retroviruses. They infected us as RNA viruses, made DNA copies of themselves, and inserted themselves in our genome at various places. We have shut down their transposition around the genome, but we still have them. Many of them are not working, but some do occasionally. So, but we do not normally produce reverse transcriptase. It would be much simpler just to go and get the gene from HIV, and then you can produce this enzyme. So, I haven't looked for it, but I bet if you go to any of the biotech websites, you can just buy reverse transcriptase. Then you'd mix it with your messenger RNA, and you'd be good to go.
Once we have this, now it's in the bacteria. How do we get the insulin the growth hormone that's producing this back into us? Like, how do we extract it from the
So how do you get proteins out of bacteria? So that is mostly done with what are called affinity columns. So you have to burst the bacteria open in a fairly gentle way. You then run the extract over a column that binds proteins, and then you wash the proteins off with different salt solutions, and then you have to be checking for the one that you want. It's not exactly simple because the bacteria contain tons of other proteins that you don't want.
Okay.
So yeah.
One more quick question. The plasmid that gets inserted into the bacteria uptake it through transformation as well.
How would you get the bacteria into the plasmid into bacteria? Yeah, that's transformation, right? So these are these processes that were discovered long ago, right? Transformation is an old thing. Understanding how the lac operon works is a long time. And so people have just adapted what we know to make use of it in the biotech industry.
Yeah.
But that's where I'm going to end, and I will see you on Tuesday.